Thank you. Thank you for the nice introduction and the invitation. So today I'm very happy to introduce our recent progress in developing quantum algorithms to study to conduct metabolic analysis and using elementary mode analysis. So my lab has a long-term interest in studying metabolic reprogramming diseases. So the metabolic changes happen in almost all disease types, especially the chronic disease, and understand them help us to better understand the underlying disease characteristics, enable us to identify new drug targets and discovery of metabolic biomarkers, and also we have a long-term goal to provide personalized nutrition and diet recommendations. I want to note here that to study metabolic changes, the most important measure is metabolic flux rate, which characterizes the reaction rate of each reactions in a specific context, such as a disease, a patient, a tissue, organ, a cell type, or even single cells. And the key question is that how to quantify metabolic flux rate in different disease and other contexts. So right now, there are a few classic approaches, and using the just a, we call it classic computing approach to derive metabolic flux using omics data. One type of this analysis, one example is used based on flux balance analysis, which utilizes linear programming to go through the flux balance condition, which describes that at steady state, the influx and outflux of intermediate reactions are balanced. So basically, this type of method identify by kind of the solutions satisfy flux balance condition and also have bias alignment with the observed omics data. And recently, my lab developed another approach which based on the physics-informed neural network. And the underlying idea is to use a neural network to characterize the nonlinear dependency between observed data and the flux. And and also during the training of the model, we incorporate flux balance and other conditions like kinetic properties as constraints. So the underlying idea of all of these methods is to integrate or leverage the goodness of fitting to the observed data, which is almost always static and with additional constraints like flux balance constraints. So here we have a real world analog like these photos. It's are static data, but it can tell us certain information about the traffic flow, similar for the metabolic flux analysis. So with recent applications, I really identified the demand of more scalable and flexible flux analysis. So one case is that to better characterize disease-specific metabolic change and identify disease-specific biomarkers, it's necessary to build a reference map of human metabolism of normal human cells. And before the availability of experimental fluxomics data and also metabolomics data, the most available data or complete data of a pan-human tissue normal cells is the gene RNA, I think it's RNA data. So recently, my lab created more than 5 million cells of healthy cells from multiple consortium, such as tabular sapiens, human cell atlas, and other consortium. And by applying the flux analysis tool to this data, and we can see distinct heterogeneity even among the cell of the same cell types from the same patient and from the same organ. So this figure shows the t-SNE plot derived from the predicted flux of pseudobulk data of tabular sapiens. We can already see the kind of the intercellular metabolic heterogeneity of the same cell type. And similarly, after another, we also identified another necessity to improve the method. So for the at single cell level, individual cell level, the cell unnecessarily may satisfy the flux balance condition, as single cells may form the like energy consumer, certain cells may contribute certain substrate. So it's necessary to develop a more flexible method to get better query of the neighborhood of a flux balance solution space rather than itself. And also, we know that the probabilistic model enabled like by MCMC is always favorable for this type of analysis because it offers more robust and comprehensive characterization. So all of this drove us kind of the consideration of a more scalable approach. But we want to know that using the current flux analysis method for the most kind of top performing one, so to analyze about 50,000 cells, it may take about around 1 day. So Then with the progress of the quantum computing, we consider if we can utilize the quantum computing to benefit this analysis. So we start from old but the simple metabolic modeling consideration, which is called elementary mode. So basically, the elementary mode describes the simplest and non-decomposable pathways that satisfy flux balance condition. So mathematically, we can tell is we can characterize as this. So define a reaction network, and with this stoichiometric matrix SIs, which describes the input, and output of each metabolite in each reaction, and all the elementary mode satisfies the conditions that S multiply V equal to 0, and V from the V is a binary variable, V is a binary vector. So, and also there are some additional constraints like simplicity, but for the for simple illustration, we just use this definition. And during the recent actually quantum computing innovation lab, at that time I start to interact with quantum computing scientists, and at that time we developed the first formulation using Grover search. And oh sorry, the figure is missing, but basic idea is that if we can incorporate the S multiply V equal to 0 into the Grover search formulation, and to identify the potential solutions. But after that, after we came back, we try to find other solutions, and we found that the QUBO really kind of perfectly fits this question. So as shown here with the QUBO formulation, so the objective is to minimize the quadratic form of this S multiplied V, and S is pre-given of the S stoichiometry matrix, and V is to be computed by elementary mode and with binary values, and we can add additional constraints that satisfy the formulation of QUBO, and with leverage the goodness of fitting of the data to the sampled or computed elementary modes. And to solve the QUBO, we have two approaches. One is using QAOA circuit, and the other is using the directly using D-wave quantum annealing. So in this today, I want to talk about some progress we achieved from this study. So first, as this is a real-world application of quantum computing, we want to first prove the concept to demonstrate if quantum algorithm can truly solve this problem. And second, we want to provide theoretical derivations and support about the quantum advantage, sampling advantage, and computational advantage of this question. And also, we will further tell about the performance and robustness evaluation, and also right now our progress in developing this into a practical computational pipeline that can be applied to 5 million human cell pool. So we start from some theoretical analysis. We work with Dr. Feli from George Mason University, who is a theoretical computer scientist. And in short, this slide just demonstrates that. So with adopting certain theories, we found that Grover search doesn't offer scalability advantage compared to classic solutions. And similarly, Dr. Feli also derived that QAOA doesn't have a kind of a scalability advantage here. But when we check the QUBO solutions, so in practice, QUBO quantum annealing can be more efficient than classic methods. And the reason is that the quantum tunneling enabled by the system directly enables the system to break through the energy barriers, which means it is more efficient to move from the local minimum and to reach the global minimum. But however, based on the current technology and theoretical limitation, so we cannot have a fully derived theoretical advantage of it. But in practice and intuitively, it may work and have a based on the nature of the computational nature, we consider this may have a certain advantage. So then we implemented the methods. So the Grover search, we use the IBM simulator quantum simulator to implement it. And for the QAO-based solution, we implement on IBM simulator and also IronQ system. And for the QUBO annealing, for the quantum annealing, we directly utilize the D-Wave, we license the D-Wave service. So for the Grover search, we found that because we use the IBM simulators, we start with some toy models rather than the real world network, which have smaller network size and simpler network size. And we consider with kind of on this system, if it works, it may suggest that we can go to the next step. For global research, we found that the kind of on simple toy models, we found that the kind of if we tell the global research what the solution will be, as straightforwardly as expected, it will generate it. But if we implement the loss term into the global search, it doesn't work. And for the QAOA based solution on the IBM simulator, it kind of the results are quite kind of randomized. But with on the iron Q system, we found that sometimes the expected elementary mode can be identified, but still there is suffer a robustness issue and also some with a high false discovery rate. And on the D-Wave quantum annealing machine, so we found that with this toy model, we found it works. So here, for this toy model, contains 6 compounds and 10 reactions with 4 expected elementary mode. We found that the Kubel, the Kubel question solved by D-Wave really kind of outputs the expected elementary mode with high percentage and plus a 0 trivial solution. So we also check the energy, which is defined by the imbalanceness level of the called result. Because for a certain result it called, it will have a kind of, it's not a true elementary mode, but have a certain certain imbalanceness. And we found that the lower imbalanced ones have higher percentage as the output. So then we benchmark it on different network shapes and different network size. We found on the chain reaction and chain perturbations, we can see that when the network size increased to a 20 or above, the percentage of the here y-axis shows the percentage of the output with 0 energy, and we see this kind of the true result percentage decrease. And for the grid shape ones, also similar pattern. For the tree shaped reactions, we get better tree shaped network, we get better performance. And also we tested it on the small world randomized network we found, which is closer to the real world question, with also see similar patterns. And after the network size increased to 25, and the performance drop. Also, we tested if we can change the Kubel loss term, and by adding additional considerations to improve the performance. For example, if we constrain the output elementary mode be with more reactions to regularize this, and we see the performance increase for certain networks, but not all of them. And we also conduct the analysis under different settings using different number of shots. And to answer the previous question about the reproducibility, for this task, I think the reproducibility is good. So we can see that the kind of with 10,000 with 1,000 shots, we can see that still the energy of the 0 energy percent elementary mode get most kind of highest occurrence percentage, but and also the lower the resolution of lower energy get kind of a higher percentage. So same for the grid shaped network. So we want to move this to the real world analysis, real world question. And when we move to real world questions, the challenge is the real world network is big. And so we need to test the different network decomposition or simplification methods, and including DW decomposition and network partition methods. Currently, I think all the methods we can achieve substantial elementary modes and good performance in the decomposed part. But right now, we're lacking of an effective approach to combine the EMs derived from decomposed modules and to get the complete EM. This is what we are working on. And on the other side, we consider another approach with using hybrid annealing strategy. We consider as the quantum annealing, which the strength is to escape the high and narrow energy barriers, but the classic simulated annealing can cross wide and shallow barriers. So we utilize the quantum annealing first and to get certain results which are not super balanced, and then utilize the classic simulated annealing to refine it to generate elementary modes. So we tested on a real-world data set called glucose glutamate metabolic network, and which contains 30 reaction modules and 15 intermediate metabolites. And we utilized the idea of annealing first, and followed by simulated annealing, and then we get kind of we apply it to we identify the 430 distinct elementary modes, and also applied and when we apply it to the tabular sequence data and using this generated EM to do another round of weighted sampling to get the flux distribution, we can see that the distinct cell type specific metabolic distribution as shown in this T-SNP plot. And also when we check the detailed metabolism, we found kind of the result quite aligned with the common sense, like the erythrocyte has the lowest metabolic rate, whereas the activated T cells, macrophages, myeloid cells, and the smooth muscle cell, endothelial cells have a higher level of metabolic rate. So to conclude, so we demonstrate that the quantum annealing-based QUBO provide computational advantage to do further data-driven metabolic flux analysis, and the main advantage is led by the sampling capability, and also especially it can generate a certain result with imbalance, which is actually the desired for this type of analysis. And also the existing QUBO laws achieve robustness performance when network size is smaller than 25, which means for the real world data analysis, we need to have a better decomposition way. And in the future, we want to evaluate whether the sample distribution to match the real world flux data, and we want to leverage QUBO with network decomposition in real-world data analysis to make better real-world analysis, and also want to design quantum-inspired algorithm by integrating quantum algorithm and classic algorithm to solve this question better. So at last, I want to thank my student, Dr. Mi Yang, who is a second-year student at OHSU, hands-on work, and my collaborator Fei Li, Ping Ma, and Yong Chen, and my funding support. Thank you.
Thank you, Dr. Zhang, for the great application by using QUBO on a real biomedical problem. So we have time for one question.
Hi, thank you for the talk. I have a question. In your summary, you said it's working robustly for networks smaller 25. So I think you're talking of degrees of freedom, or 25 what?
25 reactions. 25 reactions. So in our model, we formulate each reaction as 1 qubit. So kind of the reaction, if the reactions, the network size increase, number of reactions increase, the result will be less robust.
Okay, can I ask a follow-up?
Yes, please.
I was just wondering, because state-of-the-art algorithms so far like EFM2 and MPLRS, which is very parallelizable, and I think it works up for networks up to like 120 or more reactions so far. And I was wondering, how would you expect it to scale? Like, what would be feasible to do due to the high degeneracy of metabolic networks? Because right now, GEMS are like totally infeasible to do, and whether you would expect that you could do this at some point, or if it is like not reproducible anymore then.
Okay, very good question. So I think the key issue here is the goal of the analysis. So here, our goal is to get a reference map of the human cellular level metabolic landscape, and we want to get for each cell generated its metabolic flux distribution. So, and the ultimate goal definitely is to conduct a cell-wise analysis. But before the cell-wise analysis is ready, we think is before the method really reach scalability, to reach such scalability to enable this analysis, we think the best approach is to utilize a certain computational approach, including the quantum computing or either classic computing, to comprehensively generate the elementary flux mode and expanding it to less kind of to less rigorous definition, like with certain imbalanceness, and then conduct a weighted sampling and using the expression of each cell. So to do this, so I think the kind of because this the idea is to get the distribution of the flux at kind of a theoretical condition, and then using weighted sampling to generate it. I think it's a highly reproducible, but the key issue is about the scalability here. So this is what we are trying to do. Yeah.
Okay, so you're like trying to sample essentially then, and stepping away from the like complete elementary mode representation.
Yeah, exactly. Yeah.
Oh, okay, thank you.
Thank you.
Okay, thank you, Dr. Zhang. Also, thank you for answer the reproducibility question.
Thank you. This is what we see. Yeah, thank you. Yeah.
Thank you. Bye. So our next speaker is